Hello everyone, welcome back to another video, that is Developmental Biology Important MCQs Part 2 for Set Net Life Sciences by K&K Knowledge Hub. See the first question. In a seed, which of the following represents saprophytic tissue? Option A, testa, option B, cotyledon, option C, endosperm, option D, hypocotyle. The answer is option D, that is hypocotyle. So, saprophytic tissue refers to the diploid tissue of the plant that gives rise to the embryo and eventually mature saprophyte, that is main body of the plant. In a seed, hypocotyle is the part of the embryo located between the radicle, that is embryonic root, and the cotyledons, that is seed leaves. So this hypocotyle plays a role in the early development of the seedling after germination and is derived from the saprophyte generation. Next question. Which of the following are true for telolithal eggs? So option A, more yolk concentrated at vegetative pole, more concentrated at animal pole. They are fairly small in size. They have little amount of yolk. So the answer is option A, more yolk concentrated at vegetative pole. So here in telolithal eggs, what happens means yolk is mostly concentrated at one pole of the egg, known as vegetative pole. So vegetative pole is the opposite pole from where the animal embryo develops. And this arrangement allows the animal embryo to be positioned closer to the surface of the egg, where it can receive oxygen and nutrients more effectively during the development. See the next question. In which of the following amnion is not present? Option A, amphibian. Option B, reptiles. Option C, aves. Option D, mammals. This is option A, amphibians. So, amphibians do not lay amniotic eggs and they have a different mode of reproduction, and their eggs are typically laid in water or in moist environments. Amphibian eggs lack extraembryonic membranes like the amnion, chorion, and allantois, which are characteristics of amniotic eggs. Next question. Extraembryonic membranes are option A, not providing food for the developing embryo. Option B, certain membranes which enter in the formation of embryo. Option C, having no functioning in developing embryos. Option D, certain membranes which do not enter in the formation of the embryo. Option D, that is certain membranes which do not enter in the formation of the embryo is the correct answer. So why they don't have any entrance towards the formation of embryo means, so extra embryonic membranes or specialized membranes that are essential for the development and survival of the embryo in amniotes, which include reptiles, birds, and mammals. These membranes surround and protect the embryo in the amniotic egg or in the uterus of mammals. So, extraembryonic membranes include amnion, chorion, yolk sac, and allantois. So, these membranes are distinct from the embryo itself and do not contribute directly the formation of embryo's body structure. And next question, which of the following is breaking the sequence of development? Option A, morulation. Option B, neurulation. Option C, blastulation. Option D, gastrulation. Option B, neurulation is the correct answer. So, neurulation is a subsequent stage that occurs after gastrulation in the development. During neurulation, neural tube is formed, which eventually gives rise to the brain and spinal cord of the developing embryo. The neural tube arises from a specialized group of cells called a neural plate, which folds and fuses to form a hollow tube. This process occurs after the establishment of three germ layers during gastrulation. So the development of nervous system is a crucial milestone in the embryonic development of vertebrates. Next question. The activation of sperm in mammals is known as option A acrosome reaction, option B capacitation, option C fertilization, option D cortical reaction. Option B is the correct answer, that is capacitation. So capacitation is the process through which the sperm become fully functional and capable of fertilizing an egg in mammals, including humans. After ejaculation, sperm need to undergo capacitation within the female reproductive tract to acquire the ability to fertilize the egg. This is nothing but capacitation. See the next question. The correct sequence of gastrulation in frog is. So here they have given the order. So now we need to see what is the correct order here, like 1 or 2 or 1 or 3 or 1 and 4, like that. So first is involution, second one epiboly, third one invagination, fourth one convergence. Option A, B, C, D they have given. So let's see what is the answer. Option C is the correct answer, that is 1, 2, 3 are correct. Involution, epiboly and invagination are correct. So gastrulation is a fundamental process during embryonic development that involves the movement and rearrangement of cells to form the three primary germ layers. Those are ectoderm, mesoderm and endoderm. The correct sequence of gastrulation in frog is involution, epiboly and invagination. So next, sperm lysine include, first one is hyaluronidase, second one acrosin, third one pepsin, fourth one antifertilin. And they have given three options here. So we need to select one option. Option A is the correct answer. 1 and 2 are correct. So hyaluronidase and acrosin is the correct answer. So sperm lysine include hyaluronidase and acrosin. So sperm lysins are substances or enzymes that sperm use to penetrate and dissolve the protective layers surrounding the egg during fertilization, enabling successful sperm-egg interaction and fertilization to occur. See the next question. The term grey crescent is associated in the region of the egg of animal pole, vegetative pole, frog, chick. So what are the answer? Answer D, that is 1 and 3 are correct. Animal pole and frog are correct. So generally in frog eggs following fertilization a special region called grey crescent appears, and this region is formed on the side opposite to the point of the entry of the sperm. So grey crescent is a critical development landmark in frog embryogenesis as it marks the dorsal side of the future embryo. The presence of grey crescent is a result of cortical rotation, a process that occurs during early embryonic development in frogs. Next, acrosome reaction in sperm is initiated with option A capacitation, option B fertilization, option C influx of sodium in sperm, option D release of lysins. Answer is option C, that is influx of sodium in sperm. So initiation of acrosome reaction in sperm is triggered by the influx of sodium ions into the sperm cell. The acrosome reaction is a calcium dependent process, and the entry of sodium ions into the sperm is an essential step that leads to the subsequent influx of calcium ions. See the next question. The process of differentiation of spermatozoa from a spermatid is known as spermiogenesis. That is option A. Spermiogenesis is the correct answer. Spermiogenesis is process of differentiation of spermatozoa from spermatids in the testes of male animals including humans. See the next question. Prevention of polyspermy of an egg predominantly contributed by a process called as option A, acrosome reaction, option B, cortical granule breakdown, option C, membrane receptor activation, option D, inactivation of sperm. Option B is the correct answer, that is cortical granule breakdown. During fertilization, when the sperm fuses with the egg's plasma membrane, it triggers a series of changes within the egg. One of these changes involves the cortical granules, which are small secretory vesicles located just beneath the egg's plasma membrane. Next question. Release of calcium from the ER in a fertilized egg is triggered by option A, MAP kinase pathway, option B, inositol triphosphate pathway, option C, WNT signaling pathway, option D, CCAP depending signaling. Option B is the correct answer, inositol triphosphate pathway. So, when the sperm fuses with the egg's plasma membrane, it initiates series of intracellular signaling events. One of the key events includes the activation of phospholipase C, an enzyme that hydrolyzes phosphatidyl inositol 4,5 biphosphate into two important second messengers, that is inositol 1,4,5 triphosphate, that is IP3, and diacyl glycerol, that is DAG. Next question. Which of the following statements about fertilization is wrong? Option A, it restores diploid chromosomal number. Option B, it results in the formation of first polar body. Option C, polyspermy is prevented by the zone of reaction. And option D, it determines the genetic sex of the zygote. Option B, that is, it results in the formation of first polar body, is the correct answer. Fertilization does not result in the formation of the first polar body. Rather, it leads to the formation of second polar body. During fertilization, when a sperm fuses with the secondary oocyte, it triggers the competition and of the second meiotic division. This results in the formation of second polar body and the release of a mature haploid ovum. Next question. What acts as an inductor in animal embryonic development? Option A, chorda mesoderm, option B, mesoderm, option C, ectomesoderm, option D, endomesoderm. So, correct option is A, that is chorda mesoderm. So, chorda mesoderm is a specialized region of the mesoderm that is involved in the formation of the notochord. The process of induction refers to the ability of one group of cells or tissues to influence the fate or differentiation of neighboring cells or tissues. In this case, the chorda mesoderm acts as an inductor as it induces or influences the development of the notochord in adjacent cells. See the next question. Which of the following is primarily responsible for hydrolytic enzyme activity during sperm-egg interaction? Option A, ZP1, option B, acrosin, option C, spermiase, option D, glycolipase. Option B is the correct answer, that is acrosin. So, hydrolytic activity of acrosin is critical for the sperm to successfully penetrate the protective layers of the egg, leading to the fusion of the sperm with the egg, and ultimately fertilization will take place. Next question. Double fertilization is a characteristic feature of option A, dicots, option B, monocots, option C, gymnosperms, option D, both dicots and monocots. Option D is the correct answer, that is both dicots and monocots. So, the unique process of double fertilization is the defining characteristics of angiosperms, that is flowering plants, and is observed in both dicots and monocots, which are the two main classes of angiosperms. Next question. Which of the following sequence is correct for vertebrate development? Here we have the sequence of blastula, gastrula, neurula. So let's see what is the correct option. Option A is the correct answer. Blastula, gastrula, neurula. So odd number of cells. Sequence represents the chronological order of these three major stages during early embryonic development in vertebrates. See the next question. Odd number of cells during cleavage is found in option A mice, option B frog, option C fish, option D snake. Answer is option A, that is mice. So odd number of cells during cleavage are found in the embryo of mice due to asymmetric cell division that lead to the formation of daughter cells with unequal sizes. It's important to note that the number of cells during cleavage is not odd throughout the entire cleavage process in mice. Instead, odd number cells may be observed during specific divisions. So this is about today video. So here we have covered some important questions regarding developmental biology. In the next video, that is part 3, we are going to discuss some more questions regarding developmental biology. Hope you like this video. Please subscribe our channel KNK Knowledge Hub for more videos. Thank you.